So, in this model I will discuss that the rationale for the development of the hydroxyapatite zinc oxide composites. So, particularly this zinc oxide. So, this zinc oxide is known for this antimicrobial antibacterial properties. So, these antibacterial properties has been utilized here to develop hydroxyapatite zinc oxide composite. So, one of the things that I must mention at the beginning of this module is that there is something called antibacterial versus cell viability. I do not use the term antimicrobial simply because microorganism means that that bacteria, protozoa and all other microorganisms. So, but here we are discussing only on the antibacterial properties of hydroxide zinc oxide composite. So, the idea is that one can add the zinc oxide to an optimal amount, so that it will cause antibacterial or it will induce antibacterial property or bactericidal property. Now, bactericidal property and bacteriostatic property, this has been mentioned in one of the earlier modules. So, here we will see that what is the bactericidal property because of the addition of zinc oxide, but at the same time the zinc oxide addition should not inhibit the cell growth on the substrate or should not inhibit the cell functionality on the substrate. So, that is very critical, because the moment you start adding the silver or zinc oxide, it may potentially cause toxicity to the eukaryotic cells. So, it may kill the prokaryotic cells, but it may cause toxicity to the eukaryotic cells, and therefore, eukaryotic cell viability will be disturbed. So, we will show that how zinc oxide content needs to be tailored while developing hydroxide zinc oxide composite, so that it will induce antibacterial property, at the same time the cell viability property is not compromised. So, having said this, that this is the objective of this present module, to discuss that whether this incorporation of zinc oxide has any influence on densification, hardness, fracture toughness, how it will influence the antibacterial property on these materials, and then in terms of both the physical antibacterial properties and so on, and cell viability properties. So, just to review some of that earlier discussion, that is that we develop several biomaterials, or for a matter of fact that worldwide that a large number of biomaterials are investigated for orthopedic applications, for dental applications, for other general for cardiovascular applications and so on. But all these development the development of all these materials are inspired to address certain issues, and these issues include, for example, cytotoxicity or biocompatibility, host response, or antibacterial property, resistance against infection and biofilm formation. So, hydroxyapatite is a primary choice, as you know that it has been mentioned in this particular NPTEL course that hydroxyapatite has the inorganic composition of the natural bone. So, therefore, hydroxyapatite has been investigated quite widely. But some of the major limitations of hydroxyapatite is that it has very low fracture toughness, it has absence of antimicrobial property and limited contact with host tissues. And why zinc oxide? Because zinc oxide, like MgO, CO, it is increases the pH of the environment, which may be detrimental for the osseous tissues, and it can produce H2O2 in the culture conditions. So, just to recall what is bacteria. Bacteria is a unicellular prokaryotic microorganisms. It is very small in size, like 1 to 4 micron in diameter, and there is different separate bacteria, but out of that the most pathogenic bacteria which can kill which can cause prosthetic infection is Staphylococcus or Staphylococcus epidermidis. Most widely investigated bacteria is Escherichia coli for most in the context of the biomaterials applications. Now, these things has been mentioned very critically and sufficiently in earlier lectures on the bacteria, that structural characteristics wise it is the cell membrane structure which is distinctively different between that gram positive and gram negative bacteria. And in case of the gram negative bacteria, it is a triple layer structure. You have an additional intermediate layer of the peptidoglycan layer, which is between the two outer membrane and the inner membrane, whereas this cell wall of the gram positive bacteria, you have the peptidoglycan layer is very thick and you have that additional cell membrane as usual. Now, coming to the biofilm formation, which starts with this colony formation of the ability of a colony formation of a single bacteria. Now, once this colony goes and then it makes that micro colony, and then once it grows, then it is essentially, it can potentially form this large stable biofilm. And this biofilm you can also investigate using scanning electron microscopy, just to see that it is like a spherical bacteria, which is spherical Staphylococcus bacteria, which is pathogenic bacteria, and when it forms this biofilm, then it spreads all over the place, and then it forms a more like extracellular matrix like features, and which will help adherence of the numerous bacteria on a material substrate. These the depending, since it is very important, this biofilm formation, this has also been published in some of the high impact journal like Science in 16 to 17 years ago by Costerton and his group. Now, hydroxyapatite zinc oxide, it has a reasonably good density because this is all conventionally sintered materials, it is not even spark plasma sintered. In normal 1200 degree Celsius, this material could be sintered in air. As far as the fracture toughness is concerned, this fracture toughness of pure hydroxide is 0.7, and then as you see that if you keep on increasing the zinc oxide content, fracture toughness does not increase significantly to a greater extent, but to a reasonable extent. 0.7 it goes to up to 1.5 or 1.7 MPa square root meter. Now, when you see the distribution of the zinc oxide in the original unsintered conditions as a particulate, it has a more like needle like shaped morphology. Now, when you do this needle like shaped morphology, if you add to hydroxyapatite, then during sintering it grows at a more like a equiax shape, and this has been confirmed from the EDS analysis that indeed that equiax shaped morphology is that zinc oxide particles. Now, antibacterial property of this zinc oxide composite. So, you we have one can use the standard methodology, like you start with that polishing of the sample, then you do surface roughness measurements, cleaning, sterilization, then washing by PBS, and then finally incubation. And then incubation it goes like in 37 degrees Celsius in a normal ambient atmosphere, and then subsequently you do turbidometric analysis just to quantify that what is the bacterial viability in growth medium. Now, in terms of the antibacterial viability on the hydroxyapatite zinc oxide composites, when you grow that E. coli, that is, as I said, that it is widely used bacteria, it has a very rod shaped morphology, and as you increase the zinc oxide content, and this way it is increased. Okay. So, as you increase the zinc oxide content, you see at the highest 38% zinc oxide hardly you see any bacteria, but at the same time you see certain features on the material surfaces which indicate that zinc oxide essentially is leached out. So, zinc oxide is leached out and causing the bacterial death, and suddenly once it increases the zinc oxide, you see that colony formation ability is also certainly reduced, and then bacteria is more like isolated conditions, not like in a colonized form. Now, as I said in one of the earlier module, that any type of cell related study or bacteria related study must be conducted using not only one strain, but with multiple strain. So, that cell strain type dependent behavior can be assessed more appropriately while assessing that biocompatibility or antibacterial property or cytocompatibility property of this material. Now, in the context of bacteria, what is recommended is to use some 1 or 2 strains of the gram negative bacteria, 1 or 2 strains of the gram positive bacteria. So, that relatively moderate spectrum of the bacterial strains are investigated to see their, to assess their viability, to assess their biofilm formation ability on any biomaterial substrate. In this context, we have also done, since this is from our published results and essentially the TEMTech work of Naresh Saha at IIT Kanpur, when I was at IIT Kanpur. So, we have used Staphylococcus aureus bacteria in addition to E. coli bacteria, and Staphylococcus as expected from the morphology, it is spherical shaped morphology, spherical morphology, and as you see that as you increase that zinc oxide content, certainly less and less number of bacteria is seen on the SEM image. And these also, it is not many times in a very clustered conditions, but maybe 1 or 2 bacteria together. So, colony forming ability and then number of bacteria which is still adhering on the material substrate certainly is reduced by the addition of zinc oxide, and then we have assessed up to 38% zinc oxide conditions. So, not only Staphylococcus aureus, that is the pathogenic bacteria, another bacterial strain which is gram positive, that is Staphylococcus epidermidis, and is in the Staphylococcus epidermidis also we have established or we have seen similar observations. But here again I would like to show your attention, draw your attention that some of the places you see that zinc oxide is leached out, okay, and this zinc oxide is leached out and then it leaves certain impression behind that shows that zinc oxide is leached out and causing less bacteria to adhere and survive on a bacterial substrate, and you hardly see 1 or 2 bacteria, or this bacteria is often one bacteria and it is in the dividing stage. So, if you go closely and see that higher magnification image of this bacteria, you will see that bacteria 1 to 2 that binary fission is taking place, and in the process it causes some furrow at that or cleavage in that bacterial morphology. So, this is that CFU plate count. What you see that pure hydroxyapatite colony numbers is 73 and it significantly it is reduced to less than half in the 29 or 30 in case of when 30% zinc oxide is added. So, if you now plot this 73 to 29, it certainly shows that if you plot CFU versus zinc oxide content, so certainly it is reduced like this, almost like systematically, may not be in a linear manner, but certainly it will show systematic reduction in the CFU count with the addition of the zinc oxide to hydroxyapatite. Now, what is the role that zinc oxide plays in causing this antibacterial property? That is one of the role that zinc oxide plays is the H2O2 formation. So, essentially what is expected is that cell death is caused by the decomposition of the cell wall followed by subsequent decomposition of the cell membrane. So, essentially cell membrane integrity, which I have mentioned while discussing some of the antibacterial assay. So, cell membrane integrity is disturbed by this zinc oxide addition, and some of the potential reaction that I have seen that if you see that it essentially forms that H2O2, and this H2O2 is one of the ROS species which can penetrate to the cell membrane and kill the bacteria. In fact, H2O2 treated cells are often used as a control which will definitely show some cell death. Okay. Now, this statement can be substantiated by using some of the published literature, not from our group, but from elsewhere. For example, Briner and team published a paper in Nanoletters in 2006, where they have reported that how E. coli bacteria can be killed in the thin section, but that their morphology can be disrupted quite significantly by addition of zinc oxide nanoparticles. Similarly, another team has also shown that TM image of the zinc oxide nanorod, and then how TM and then very thin slice of the bacteria, particularly E. coli and some of the other bacteria B. atrophius, and this bacteria also shows that there is signs of the membrane disintegrity, as you can see that there is more poration in the membrane. So, once this membrane disintegrity is lost, membrane disintegrity is taking place, then bacteria cannot survive. Essentially, all the cytoplasmic material, organelles or cytoplasmic material can come out and causing the bacterial death. This is another work by this E. coli just to show the TM image of the bacteria treated with H2O2, and here one can confirm that how this bacterial morphology also is disturbed or disrupted because of the H2O2 treatment. Now, as I said that antibacterial property, all this is fine, but at the same time one has to find out that what is the cellular viability or cell viability on this hydroxyapatite zinc oxide composite. So, not only zinc oxide addition should cause the antibacterial property, but it should not disrupt the cell viability properties because of the zinc oxide addition. Here one has to show very carefully. Now, let me write down this. D essentially means that 5% zinc oxide, 5% zinc oxide. So, this is essentially 5% zinc oxide, and you see that there is certain morphological changes that is started taking place from 5% onwards, and then this is the 30%. These 30% cells are spherical in nature, which certainly shows cell does not cell does not undergo any morphological alterations, or in other word cell morphology remains spherical while they are even being attached to the substrate. So, that shows that kind of cells are not adhering and expanding, and that is the kind of indication that cells are perhaps are not viable. Now, one has to quantify all these viability things using that MTT assay, which I will show you later, but before that I will also discuss with you that how these L99 mouse fibroblast cells, they adhere and proliferate to some extent on lower zinc oxide content materials, that is 1.5 and 5.8%. Once you add 20% zinc oxide, here again it does not show very regular spindle like morphology like a fibroblast cell should exhibit, like in this case and this case. So, certainly you can say that 20%, 30% would not favor the cellular response the way lower zinc oxide content materials they would essentially show. Now, same thing we have seen also when you use that sarcoma osteosarcoma cells, that is human osteoblast like cells, and you see that 1.5, 5 and 7.5% materials that show this kind of typical cellular morphology. 20% still okay, not bad, but 30% certainly is not good. So, 30% zinc oxide does not show any good cell response properties, and this is kind of reflected here in the MTT assay. So, the first one is that mouse fibroblast cells, that is L929 cells, which is the connective tissue cells, which is widely used biomaterials research. You see that there is a progressive systematic decrease in the cell viability as you increase that zinc oxide content in the material, and perhaps that most significant statistically significant results are obtained from 5% onwards, and 7.5%, 10% is certainly worse, 20 and 30. So, this 10, 20, 30 one should not use. People should restrict that hydroxyl zinc oxide composite at up to 5% zinc oxide. So, beyond 5% zinc oxide composite, there is a seriously systematic significant statistically significant difference in terms of cell viability with L929 mouse fibroblast cells. The similar observation to some extent or largely valid when we have quantified the cell viability using that osteoblast like cells, sarcoma osteosarcoma cells. Here you see that very clearly 20% and 30% is worst in terms of that supporting the cell viability. 10% is fine, but there are certain stars are there. So, stars means that is statistically significant results, but then when you see that with respect to control parent hydroxyapatite up to 10%, this is still okay, up to 10% one can consider, but in case of fibroblast cells you say that from 5% onwards it is not that good. So, osteoblast cells shows up to some tolerance level up to 10% zinc oxide, but overall one would recommend that cell compatibility wise that hydroxyapatite 5% zinc oxide is more safe than 10% or 20%. So, this conclude this. So, significant antimicrobial action has been confirmed using that hydroxyapatite zinc oxide using S aureus and S epidermidis strain, which is more pathogenic strain, and it was measured that with zinc oxide addition more than 7.5% indicating bacteriostatic behavior. Now, cellular responses of this material, cell attachment and flattening is observed only up to 10%. So, somewhere between 5% and 10% is the optimum zinc oxide content which can be useful for further biomedical applications, and however this thing needs to be tested in that preclinical study in the animal model, and but for that one of the useful composition can be hydroxyapatite perhaps in the middle range is the 7.58% zinc oxide additions, which shows reasonable cell viability in terms of sarcoma osteosarcoma cells as well as fibroblast cells, and at the same way it has reasonable antibacterial property. So, thank you.